previous topic, you learned about one of the DNA typing techniques. In this topic, you're gonna learn about another technique, which is called restriction fragment length polymorphism, for short RFLP. The basic premise of this technology revolves around using restriction enzymes to digest a DNA sample into fragments and to separate the resulting fragments by size using electrophoresis to produce a characteristic pattern or fingerprint. This pattern varies based on how many times the enzyme recognition sequence occurs in the target DNA sequence. RFLP technique can be used to examine restriction site variation in DNA targets of varied size. Indeed, RFLP typing was the first DNA fingerprinting method to be used to establish human identity. The basic technique for detecting RFLPs involves the following. First, fragmenting a sample of DNA by a restriction enzyme, which can recognize and cut DNA wherever a specific short sequence occurs, in a process known as a restriction digest. The resulting DNA fragments are then separated by length through agarose gel electrophoresis. The fragments are allowed to hybridize with a labeled DNA, and then the length of the fragments is determined. An RFLP occurs when the length of a detected fragment varies between individuals. Each fragment length is considered different form and can be used in genetic identification. However, there are some limitations for this technique. RFLP analysis is slow and bulky. It requires a large amount of sample DNA, and the combined process of the different steps could take up to a month to complete. Fortunately, the results of the Human Genome Project have largely replaced the need for RFLP mapping and the identification of many single nucleotide variation in that project, as well as the direct identification of many disease genes and mutations have replaced the need for RFLP disease linkage analysis. To summarize this topic, first, RFLP is a DNA typing technique. Second, one limitation of using RFLP analysis is that it requires large amount of sample DNA. And finally, the Human Genome Project results have largely replaced the need for RFLP mapping.